In the early 1800s, French biologist Jean-Baptiste de Lamarck said, "It is not the organs, that is, the character and form of the animal's bodily parts, that have given rise to its habits. It is the habits and manner of life in which its ancestors lived that fashions its bodily form, its organs, and its qualities." What was Lamarck referring to, and why did Charles Darwin paint a different picture? I am your host, Bruce Lipton, for the series on inner evolution. The notion that genes control our lives was first predicated on Newtonian materialism. Science was seeking the physical basis of evolution, the search for the genesis elements, as an insight into understanding the mechanisms that control life. This pursuit led to the Human Genome Project. In what amounted to a cosmic joke, the project's results completely undermine the assumption that genes control evolution and the character of life. The door was now open for the new and personally empowering science of epigenetics. In 1859, a theory of evolution by Charles Darwin was published on the origin of species by natural selection. I want to make a very important correction at this moment for this fact because we give credit to Darwin for the theory. The truth is, it was Alfred Russel Wallace, a commoner working in the Malay archipelago, that actually wrote the theory of evolution and sent it to Charles Darwin. Darwin was taken back by the fact that someone had already written this theory and he didn't finish his effort of writing his theory. Well, Charles Lyell, head of the Royal Society, and Darwin got together, and under what they referred to as the delicate arrangement, they put Darwin's name in front of Alfred Russel Wallace's name when presenting the theories at the Linnean Society meeting back in 1858. The significance is profound because priority is always given to the first author. Darwin took the work and within the year came out with his book on the origin of species, and Wallace lost in history. Now, the significance of Darwin writing the theory is quite different than understanding Alfred Russel Wallace's perception. Wallace, a commoner, when he saw the understanding of how animals in the world work together and compete with each other, actually came to the conclusion that evolution was based on the elimination of the weakest organisms in the community. Darwin, an upper-class person, and in supporting the upper-class philosophy, changed that around and said that evolution is actually driven by the survival of the fittest in the competition. So Wallace said it was elimination of the weakest. Darwin said it was the preservation of the strongest. They both have the same conclusion, the glass is half empty or the glass is half full, but they have profoundly different meanings. Because if I give you a chance to live in either of two worlds, an Alfred Russel Wallace world where only the weakest get eliminated, or a Darwinian world where only the strongest survive, which one would you pick? Think about it. The answer is simple. It's easiest to be an Alfred Russel Wallace because it's easiest not to be the weakest. It's much harder to be the strongest, as in a Darwin world. But Darwin was the first one to write his book on the origin of species. And since then, the world has been taken by the understanding that it is a competition for survival on this planet. And therefore, in trying to understand the nature of evolution, we must first go back to Isaac Newton. And the significance is this: in Isaac Newton's universe, the physical material realm can only be affected by other forms of matter. And therefore, in trying to understand how a human body works, and human body being a physical machine, it was understood that somewhere in that body there were physical elements that controlled the character of that machine. And when Darwin came up with this idea, there was no insight or understanding of the nature of genes. It was a philosophy that there were some kinds of structures like paint droplets scattered through the body, and that these droplets were called gemmules. And these gemmules carried the nature or character of the part of the body where they were located. So there'd be a gemule in your shoulder that would accommodate the shoulder, a gemule in your neck that would program and develop the neck. So gemules were scattered all over the body. When two people mated, the gemules of the mother and the gemules of the father would come and blend together to create the characteristic of their offspring. In searching for those physical elements, the gemules, we were looking for the genesis elements. And genesis elements can be abbreviated as genes, and that's where the term genes comes from, okay? So the significance is this. Darwin's theory was a two-step process. Step one, there's an alteration in the heredity that the so-called gemules would alter their characteristic, which in turn would alter the characteristic of the body in which these gemules were found. Followed by step two, natural selection, where nature will select the strongest of those altered organisms to survive and propagate those genes and eliminate the weaker organisms that have been altered by these genes. The concept of gemules, of course, was an elusive idea because it was just an idea, a philosophy. There was nothing real to identify with the hereditary units. But interestingly enough, at the same time that Darwin was working on the theory of evolution, in Austria, there was a monk by the name of Gregor Mendel. And Mendel was mating flowers of different colors. And what he showed is if he mated a red flowering plant with a white flowering plant, the offspring would be pink. Well, this would suggest that the characteristic in the red plant, its gemules, merged with the gemules in the white plant, created pink. So that fit the gemule story. But a whole new insight was offered when Mendel did back-crossing, where he mated two of the pink offspring together. And what happened? Not only did pink flowers show up on plants, but there were plants with white flowers and plants with red flowers. This was profoundly important because it said that whatever the hereditary elements were, they were very specific physical units. They didn't merge with each other as gemules were supposed to do. They kept their characteristics, but when present together, a red and white one would create a pink plant. But when you back-cross, any plant that received two white genes would be white, and any plant that received two red genes would be red, and the pink was the result of a plant having one of each of the genes. The point is very critical because it said that whatever the hereditary units are, they're physical, discrete units, not like paint droplets that merge, but maintain their characteristic from generation to generation. Well, that put the search on. What in the organism represents these new structures, which are now referred to as genes? What is a gene? Where is a gene? Well, in 1892, August Weismann, a German scientist in studying cells and tissue culture, observed as cells divided, there were these little filaments that were found in the middle of the cell. The filaments were split half and half, one half going to one daughter cell, the other half going to the other daughter cell. By following these filaments over and over again, he identified them first as chromosomes, and then recognized that these chromosomes were actually the hereditary units. So now there was a physical basis of where is the hereditary unit? Where are the genes? They're connected to the chromosomes. So now science was going to try to understand the nature of genes, but here's the first problem. Chromosomes are consisting of 50% protein and 50% DNA. Well, science didn't know, was it the protein that was a hereditary unit, or was it the DNA that was a hereditary unit? Well, that answer actually didn't get worked out until 1944. That was the first time science recognized that DNA was the element responsible for heredity. How did that experiment work? Well, Avery, McLeod, and McCarty did this experiment with two types of bacteria. A type R bacteria, which created little point-like colonies in a culture dish, just like little periods all over the culture dish. The other species they looked at was type S, but these formed big puddle-like colonies. And what they wanted to find out is it the chromosomal protein or the chromosomal DNA that is determining the character of these colonies. So what they did is they separated the chromosome from type S into its protein component, into its DNA component. And then what they did is they first added the protein from the chromosome to a culture of type R bacteria. The result? Nothing. It was the same bacterial colonies type R always had, the small little point-like colonies. However, the surprise came when they put the DNA from the type S chromosome into the culture with type R. Because what happened? The type R cultures were transformed to express the big puddle-like colonies of type S. It was this first experiment to identify that DNA was carrying the hereditary character in an organism. Well, now we know DNA, but how does DNA work? Nobody had any idea about DNA and its mechanism. That took another 9 years. In 1953, the answer of how DNA works was provided in the journal Nature by Watson and Crick. And Watson and Crick have been given all the credit for understanding that DNA is a double helix and encodes a gene program to provide for the characteristics of an organism. I just want to make a point right here, it's very important, that Watson and Crick were actually not the real founders of the DNA double helix. It was a woman, Rosalind Franklin, who through her research on DNA molecules first showed the picture of an X-ray characteristic of a DNA double helix. Interestingly, Rosalind Franklin's major advisor gave her research results to Watson and Crick without even telling Rosalind Franklin that he did it. It was that research that led to Watson and Crick coming forth with the famous DNA double helix picture in 1953. Well, once we started to understand the nature of DNA, we started to recognize that it is what is programming the character of an individual. It is responsible for heredity. And now comes a serious issue, because the chromosomes carrying these genes which control our character, well, we get them from our mother and our father at the moment of conception. Well, as far as we know, we didn't pick the genes that we were born with. And as far as we know, we can't change the genes if we don't like the characteristics. And a piece of research led us into another belief that disempowered us further. And that was, how is DNA controlled? Because DNA apparently is the blueprint to make the proteins of the body. Our next episode reveals that we're not victims, but we are masters in controlling our gene characteristics. Thank you for joining me. I'm your host, Bruce Lipton, for this series on inner evolution through biological change. Join me for the next exciting episode where we'll talk about how you actually control your gene activity.